In this video, we're going to take a look at how prokaryotic and eukaryotic cells move materials across membranes using passive means of doing so. So means that don't require any expenditure of ATP energy. That's what we mean by this idea of passive transport. No ATP is required. Okay, so to understand a little bit, we have to kind of make sense then about semi-permeability of cell membranes and of the plasma membrane material that we see that makes up cell membranes. So the lipids that are in the membrane, they prevent the movement of water, and anything that dissolves easily in water, so water-soluble materials, they're limited by this lipid that is in the cell membrane. And it's the proteins that allow selective materials to move through the membrane. So water does move through the membrane, but it requires a pore made by a kind of protein called an aquaporin. And so a bunch of these aquaporins make a little hole through the cell membrane, and so water can move very easily through the membrane, but only because of the pore that's made by the aquaporins. The same with ions. Proteins move ions across cell membranes, and any other water-soluble material requires a protein to be able to get that stuff into the cell. So there are two mechanisms that we see, two large groupings of mechanisms, I should say. Passive transport, the types of transport that don't require ATP energy, but instead just rely on the physical properties of materials that are dissolved in fluids to move materials from where they are in high concentration to where they are in low concentration. And we'll see that that is described as diffusion, the movement of materials by passive forces from regions of high concentration to regions of low concentration. Then there's also active transport, and that's where we use ATP material to move materials against the concentration gradient. So we activate active transport with ATP being used, allows us to move materials from where they're in low concentration to where they are in high concentration. So that's what I mean by against the concentration gradient. So passive transport is what this lecture is about. So no ATP energy is required, and because of this, we can only move materials from a region of high concentration to a region of lower concentration. And there are three passive mechanisms of moving materials: simple diffusion, facilitated diffusion, and then a special case of simple diffusion called osmosis. So let's start with simple diffusion. If we were to take, what they're showing us here is that they've taken a beaker of dye molecules and they've placed it into a large chamber filled with water. And so over time what will happen is that because there is energy in the water, that energy is expressed by molecules moving back and forth very rapidly. We saw this. The Brownian motion that you noticed in the non-motile bacteria is an example of this energy in the water being expressed as vibration. And so as the molecules are vibrating back and forth, then they bounce around and they bounce into each other. And any time that there is a collision, then the two molecules that collide, they run into each other, and shping, they go in opposite directions. So materials, because they tend to have a lot of collisions with themselves, with other, in this case, dye molecules, because they're in such high concentration here, then what happens is that as they collide, the materials are going to move away from the area where the collision happened and out away from that center of high concentration until finally everything is evenly dispersed. So at this point, it's even. The concentrations of the ion, of whatever the dye molecule is the same throughout, and so diffusion can't happen anymore because there's no region of high concentration anymore, and no region of low concentration. Membranes, though, and that process is called diffusion. So materials diffuse from where they are in high concentration to where they are in low concentration. And membranes, though, can limit the diffusion of materials because in order for something to diffuse across the membrane, it must either dissolve in lipids and pass directly through that membrane, which is how things like really small molecules like carbon dioxide and oxygen, they diffuse very easily through cell membranes. And then other lipid materials, so like steroids, steroid hormones, and cholesterol, they can pass through a membrane fairly easily because they are lipids themselves, and so they'll dissolve in that oily inner layer of the membrane. Or if they can't go right through the membrane, then they have to pass through the membrane using proteins, and this is how water moves and how ionic materials like hydrogen and potassium and sodium and chloride ions, how they all move through the membranes, is because proteins allow them to do so. So diffusion is the rate at which diffusion occurs, the speed with which molecules move away from centers of high concentration, is all depends on how easily the material that's diffusing dissolves in water, or dissolves in lipid. So the rate at which we get across that membrane really depends on the solubility of the substance in lipid. And if there's high solubility, then diffusion is going to happen relatively rapidly. Also, temperature plays a role here. And the warmer the material is, then the more energy there is in the solution, the more banging around all those molecules will do, and the more rapidly diffusion occurs. So warmer solutions equal faster diffusion. This is part of why we must maintain a warm body temperatures so that we have sufficiently rapid diffusion occurring in our cells. And then last of all, the third thing that can control or influence the rate at which diffusion happens is how high the high concentration is in comparison to the low concentration region. So the greater the difference that we see in concentrations between high and low, then the faster that diffusion will occur. And so as each side of the membrane comes closer and closer to each other in concentration levels, then we're going to see that the rate of diffusion will slow until we finally reach that point at which they are equal concentrations on both sides of the membrane. So, three characteristics that will affect the rate at which diffusion across membranes occurs. First is the solubility in lipids. Second is the temperature of the solution. And third is how great the difference is between high and low concentrations. Okay, so that's diffusion. So diffusion moves materials across membranes based on those characteristics. Facilitated diffusion is really a cool thing. It happens when there are what are referred to as carrier proteins, and just make sure that we're clear, this is about facilitated diffusion. So facilitated diffusion occurs because carrier proteins are found embedded in the cell membrane. So they have a surface on the outside of the cell, and they have a surface on the inside of the cell. So we get two surfaces, and there is a way for the material to pass through. So something can happen that will let the material pass through. So the outside of this protein has the capacity to bind a molecule that needs to come into the cell or needs to leave the cell. It can go both ways. So there's the capacity on one side of the membrane to bind a molecule of interest to the cell. And as that molecule binds, then because of the way proteins work, whenever you interact a protein with another material, it changes the shape of the protein. And so when the protein changes shape, so here we have the material that is in high concentration outside the cell, it enters into the binding site. When it enters there, it changes the shape of the protein. And now this area that was closed opens up just because the molecule bound in there at the binding site. And when it changes, when the protein changes shape, then it releases the bound molecule into the cell, or if this was happening the other way, if we're moving something out of the cell, then it would release it on the outside of the cell. But on the opposite side from which it came in, then we see that material being released to the other side of the membrane. And note that no ATP is required to cause this shape change in the carrier protein. It's just the binding of the material that causes the protein to change shape. Excuse me. So facilitated diffusion works because it has that name diffusion in there. We know it has to go from a high concentration area to a low concentration area. Sorry, I just choked on a little swig of my soda here. Oh. Okay, I think I'm better now. I think I might survive this one. So let's see where I was. Oh yes, because it's facilitated diffusion, then it's moving materials from the high concentration side of the membrane to the low concentration side of the membrane. And the speed with which facilitated diffusion can happen is really limited. You know, the amount of material we can move from one side to the other is really just limited by the number of carrier proteins that are found in the membrane. So if we want to move more material, then we add more carrier proteins until all those carrier proteins are engaged, and then once again the only way we can speed it up is to add more carrier proteins. So the cell can control how rapidly it moves materials into or out of the cell by changing the number of carrier proteins that are doing that movement and that are found embedded in the membrane. Okay, so there's facilitated diffusion. Once again, movement from high concentration to low concentration, but this time with a carrier protein that can do that can move that material across to the other side. Okay, so here's the last passive process, and it's called osmosis. Now, salts and proteins, they do not move directly through the membrane. They must move using some sort of carrier protein to do it. But water can move freely. Even though oil and water don't mix, it can move freely because all plasma membrane has these protein channels in it called perforins that allow water to zip from one side of the membrane to the other. So if the salts can't move, if what we normally think of as the solute can't move from its region of high concentration to its region of low concentration, well then the solute can move. And osmosis then is just the diffusion of water from a region of high water concentration to a region of low water concentration. And so this is a concept you should have picked up already in either BIO275, 163, or 168. So this is something that hopefully you've learned already, so you're just learning it the second time. So notice we have this U-shaped tube, and there's a membrane here that mimics the plasma membrane. It lets water through, but it doesn't let salts through. Or in this case, they're using sugars as the solute. So we have a lot of sugar dissolved on this side of the tube. No sugar, so it's 100% water on this side of the tube. So pure water, 100% water, is always going to be the region of high water concentration. Notice that where the sugar is, that's always going to be the region of low water concentration because that sugar always displaces or moves some of the water out of the way. So a 1% sugar solution is a 99% water solution. So if water can move through the membrane, and we know it can because of these perforins, then water on the distilled water side of our tube is going to flow toward the sugary side of the tube. And so what we'll see is that there's a net movement of water from the region of high water concentration, the distilled water side, to the region of low water concentration, where the sugars are. And that will continue until they both equal out until there's an equal amount of water on both sides. Now, the thing that prevents this from actually moving so much water that it flows over is that this water has weight, and the weight is going to be pushing water back the opposite direction too. So there are forces that work against this osmosis, and gravity is one force that works against it. As well, water, just by pure, simple water diffusion, is going to be moving in this direction across the membrane as well. So any collisions that happen with the water molecules will move a little bit of water to the other side. But overall, the primary direction in which water is moving, the net movement of water, is toward the region of low water concentration. So anytime we dissolve material in water, then that's going to make a region of low water concentration. And so frequently we talk in anatomy and physiology and in health that water is going to go from where there's not very much stuff dissolved in the water, our distilled water side, for instance, to the place where the water is diluted some with some dissolved material in it. And so we use the shorthand of water follows salt because it kind of explains how this all goes down. It's not a perfect explanation, but it's a nice way to think about it. Water is always going to move to the saltier place because the saltier place is always going to be the region of lower water concentration. So the regions that have this low water and high solute concentration, they're said to have osmotic pressure. And this is a weird use of the term pressure, because usually we think of pressure as pushing away from that region that has the pressure. But this time, when it's osmotic pressure, we mean that it has a high call for water to come toward that side. So you just got to keep in mind osmotic pressure, as opposed to, say, hydrostatic pressure, like with the water hose. Osmotic pressure calls water to the saltier location, to the region that has the low water concentration and the high solute concentration. Okay, so that's osmosis. Osmosis is always going to move water from where there's a little bit of water, a whole lot of water, to the place that has less water in it. And that's why water follows salt. All right, so the last thing, and this always gets confusing when we talk about osmosis, and then we talk about comparing solute concentrations. But the book has a picture of this, and I think it's a great way to think about it. When we're thinking about solute concentrations, we think of it really always as the concentration of whatever's dissolved in the water. So when you're talking about comparing solutions to each other, then you've got to remember that we compare based on not the water, but on the dissolved solute, whatever it might be. When we talk osmosis, then we're not talking about the solute anymore. We're thinking more about the water. So in an isotonic solution, iso means same. Same root as for like an isosceles triangle, which is a triangle where the two sides are of equal length. That's a terrible isosceles triangle, but you get the idea. So isotonic, tonic here means strength. So it's the strength of the solution. And if the strength of the solution on either side of the membrane is the same, then what do you think? Will water move from one side of the membrane to the other? And what we would say to that is that yes, water does move, but it moves in equal amounts in and out when it's an isotonic solution. So overall, there's no net movement of water in isotonic solutions. Water moves into the cell, water moves out of the cell, and it does it at the same rate. On the other hand, when we put cells into a hypotonic solution, oh shoot, I got these swapped. So we'll think about the hypotonic solution next, because that's the next one in the illustration here. Sorry, I should have those switched the other way around. In a hypotonic solution, the cell is sitting in a solution that has lower strength. Hypo means below. So it's a hypotonic solution because there's less salt or less sugars or less dissolved materials in the solution that the cell is sitting in than there is in the cell itself. So here, then, is a 0.5% salt. Inside of our cell is 1% salt. What's going to happen here? Now we got to think about what is the water concentration. So the water concentration outside the cell is 99.5% water, because the other 0.5% is salt. Inside the cell, though, it is 99% water. Now which way is the water going to go? Hopefully you see that the water is going to go from high to low, primarily. I'll make that a great big arrow. Water is going to flow into the cell. Now, will water flow out of the cell? Yes, it will, but only just a little bit. So the majority of movement, the net movement, is into the cell, because the saltier solution that is inside the cell is going to draw the water in, because it has high osmotic pressure. It's going to draw water into the cell. And then the last comparison is with a hypertonic solution. So here's our hypertonic solution. This time, the solute concentration that the cell is sitting in is higher, because it's hyper. Hyper means above or extra. So it's extra strong solution that the cell is sitting in. And so that means it's got lots of salt in it in comparison. So for instance, if there's 3% salt in our solution and only 1% salt inside the cell, then what we see happening is that water is going to be drawn out of the cell. And that's because in the solution we have 97% water. In the cell we have 99% water. So when we put a cell into a hypertonic solution, it's always going to move water out, because the salt that's in the solution around the cell is going to be calling lots of water out of the cell. Now, is there water going in? Think to yourself for a moment. Hopefully you're saying self. Yes, there is. In which case, yes, water also moves into the cell. We have a little bit of movement into the cell. But overall, most of the movement is out of the cell. And here we see what happens then. If we have a eukaryotic cell that is being stressed like this in a hypotonic solution, well, a red blood cell in a human is going to burst eventually if we put it into almost pure water. But will a bacteria burst? Probably not. And that's because most bacteria have a strong cell wall surrounding them. The cell wall is going to take the pressure caused by all that water flooding in, and it's going to hold it and prevent the cell from bursting. On the other hand, if we have a cell that's in really salty water, well, if it's a eukaryotic cell like a red blood cell, then lots of water gets drawn from the cell, and the cell becomes what's termed plasmolyzed, because most of the water is called out of the cell. If that happens to a bacteria, now yes, they will be affected by this. The cell membrane won't, because it's not going to be pressing against the cell wall anymore. Instead, it's going to shrivel within the cell wall. So hypertonic solutions will affect bacteria. And this is part of why we gargle with salt water, in order to help make our throat feel better, because all that gargling with salt water, getting that all around the bacteria that have colonized our throat when we have a sore throat, it's going to help to dry out those bacteria, and hopefully, if things go well, you're going to kill some of them in the process, making your throat feel better. Okay, so there it is. We've talked about all of these characteristics of diffusion as we move materials from high concentration to low concentration, facilitated diffusion, where we use a carrier protein to move the materials from high to low concentrations, and then osmosis, where water moves from its high concentration and low concentration based on how much, and that's determined based on how much stuff there is dissolved in the water. And the regions that have lots of stuff dissolved in it, then those are going to be the regions of low water concentration that are going to call water to that side of the membrane. Okay, so there you got it. You know what to do if you need me. Ask your questions, and we'll talk.